So yes, my name is Paige Maroni. I am speaking to you from the University of Western Australia on Whadjuk Noongar Boodja. I pay my respects to the Whadjuk Noongar Nation. So my talk is called Multiple Doris Cogenensis Species Span the Antarctic Polar Front. How I'm going to progress through this talk is start quite broad and then move into my study species and the results we found from this work. So by broad, I mean very broad, meaning Pangea. So here you can see the breakup of Pangea and then the subsequent breakup of Gondwana. I'd like showing this first because it gives you a really nice overview of the oceanographic and geological isolation of the Antarctic continent. What you can see is as the continents separate, the Antarctic continent begins to freeze over and is separated from Australia and South America, which resulted in the onset of the Antarctic circumpolar current. This current is today the world's largest ocean current and is powered eastward almost entirely by wind and is responsible for the transportation of water throughout all global oceans. What you can see here is a snapshot of the glacial cycles within the Antarctic region. I'm showing you the last 400,000 years, but the Antarctic has been experiencing glacial cycles such as these for the past 35 million years. And these glacial cycles are what has been attributed to such high levels of marine endemism and marine biodiversity within the Antarctic. What you can see here, for example, is an interglacial period. So the air temperature shown in blue is higher, which means the sea level is also higher because all the glaciers have melted. And then we move into Earth's last glacial cycle or last glacial maxima around 25,000 years ago. This is where you can see the air temperature has dropped, therefore the sea level has also dropped because all of the surrounding water is now locked up into ice. And today we are currently sitting in an interglacial period. So all of these glacial cycles during this time, benthic fauna within the Antarctic have been proposed to have survived either on the continental shelf in these ice-free refugia, or if the glaciers have extended out across the entire continental shelf, anything that was existing on the continental shelf itself would have been completely obliterated by these mass glaciers. And it has been hypothesized that the deep sea would have been the only area where these organisms would have been able to survive during this time. All of these environmental extrinsic factors have allowed Antarctic benthic fauna to have some very specific traits that are characteristic across a range of taxa. One being that a lot of organisms show high levels of urobathy. This means that the organisms have a very wide vertical distribution, so they can extend from 5 meters down to the deep sea, just as an example. A lot of organisms within the region also have long life cycles and very slow growth rates due to slow metabolisms. What you can see here as an example is a sea star life cycle. However, these organisms do have pelagic larval dispersal capabilities, which is common in Antarctica, but is definitely not as common as direct development. This means that the organisms just grow from being a small form of the adult into the adult size rather than going through a metamorphosis such as the sea star in the example prior. So I did briefly mention the Antarctic circumpolar current, but I really want you to remember this as an extremely complex oceanographic system. So as you can see, it's full of gyres and overturning pathways and fronts, and they're all going in different directions. And it is a very complex place, especially if you're a 3 millimeter or a 3 centimeter benthic marine organism trying to navigate through this ocean. An area I really want you to focus on is the area between southern South America, which you can see on the left, and the Antarctic Peninsula. Because although this area is by landmass the closest, what you can see is these huge big jet streams pumping through this area and into the Scotia Arc and up into the northern warmer oceans. So although this area by land is the closest and is the way that humans generally tend to try and get to Antarctica, if you're an organism, you're probably going to get pushed north before you're going to get pushed back south onto the Antarctic continental shelf. Along with this Antarctic circumpolar current, I will also be mentioning the Antarctic polar front. This front has been deemed the barrier, or is a really good example of a barrier between the Southern Ocean and the warmer Northern Oceans. And I will be mentioning it throughout the talk, so please remember this is just one front within a very complex system called the Antarctic Circumpolar Current. But for this, we really don't have many examples of benthic fauna that actually are able to cross the polar front, with absolutely no examples of direct developing benthic fauna existing on the southern South American continental shelf and the Antarctic continental shelf. A few examples of southern South America to Southern Ocean we do have include a sea star and a brittle star. Both of these organisms do have pelagic larval dispersal capabilities. We also know there are quite a few sea spiders. However, these animals are known for their extreme Eurobathism and are proposed to be able to migrate or walk across the abyssal plains. So that may be how they are getting in and out of the Southern Ocean. And then this one remarkable example from earlier this year of Tritonia vorax and direct developing nudibranch, which has been found on the southern South American continental shelf and at South Georgia Island in the Scotia Arc. So this is where my work comes in. I work on Dorus craguelensis. This animal is an Antarctic nudibranch species that has a circumantarctic distribution. It is a simultaneous hermaphrodite that lays down cross-fertilized embryos in a gelatinous matrix on the seafloor, and it feeds exclusively on sponges. Originally, Dorus craguelensis was described as a single or as a few Antarctic nudibranch species. However, by applying molecular tools, it has been shown that it is actually a multitude of cryptic species in an adaptive radiation. So for the first portion of my PhD, I was tasked with documenting this cryptic species complex. So what I did was I Sanger sequenced samples from all of these areas within Antarctica. We barcoded them all, and by using these barcoding methods as well as incorporating multiple species delimitation models, we delimited 59 species from across the Southern Ocean. For this talk, however, I'm focusing more on a select few of the species throughout this species complex. So keep the 59 in mind, but for now I'm going to tell you about just a few really interesting ones. From the phylogenetic study, we found three species that had a lot of samples within the species. And then when looking into these three very well sampled species, we found an additional three that had an extremely interesting distribution that we wanted to just get a snapshot of the idea. So here, I know a lot of you do absolutely know what a haplotype network structure is, but for anyone at home who does not know, this is a haplotype network structure. Each one of these bubbles is called a haplotype, and that represents an identical or a unique sequence. The wedges within the circles will be colored and they will be mapped alongside them. So you can see that the wedges represent where the samples are from. The hash marks in between the haplotypes represent an individual nucleotide or a base pair change between the sequences. So this can kind of give us an indication of how different these haplotypes are from one another. We were also able to look into genetic diversity indices, population demographic statistics, and genetic differentiation statistics. So these 6 species along the left here are the ones that I am going to be talking about today. The colors of the slugs are the exact same on the map as they were on the phylogenetic tree, and the size of the slug itself represents that it has more samples in it. It's a bigger slug, smaller samples, it is a smaller slug. These are all obviously undescribed as well, so we have given them numbers for the purpose of this talk and for our phylogenetic study. So I hope you can all remember, before I mentioned we have no examples of a trans-Antarctic polar front benthic direct developer. That is till now. We have 4 Antarctic nudibranch species that we have been able to find that span across the Antarctic polar front. One of these being species 24. Across the entire topology of the tree, this is the most well-sampled species we do have, which is why we wanted to look into it in the first place. This animal was found from Mount Saipool all the way to Birdwood Bank in South America, and Bouvet Island, which is the red mark at the very top of the map. Because we had such a nice amount of samples for this, we were also able to characterize the structure of the haplotype network. And for all of our well-sampled species, we have characterized the structure of these networks. So for this one here, we have classified this to be a geographically structured haplotype network, or a parochial network structure. This means that for samples that are geographically close together, they are also close together within the haplotype network structure. And we have kind of this big star-like function, but a lot of very private haplotypes throughout the network. The other 3 species that spanned the Antarctic polar front include species 14, 26, and 42. Although we could not do any phylogeographic statistics on these due to their small sample sizes, we were able to show that the unique haplotype was spanning the Antarctic polar front for all 3 of the species. Then I also had a really well sampled sub-Antarctic species. I just like to highlight that the hash line representing the polar front to the right of the slide around this Kerguelen Island plateau sample here, that indicates a newer model of the current trajectory. So we have classed this entire species to be sub-Antarctic because it is now believed that Kerguelen Island is actually north of the Antarctic polar front. So for this organism, it was also deemed to be a geographically structured haplotype network. So what you can see is, again, samples that are close to each other geographically are also close together or neighboring within the haplotype network. And finally, we have one very interesting circumantarctic species. We did have a few circumantarctic species across the topology of the tree, but this one was definitely the most well sampled. So this species was collected from over 11,000 kilometers, which for a 3 to 20 centimeter slug is extremely incredible. This haplotype network structure is very different from the other 2. We've classed it as being geographically diffuse rather than geographically structured. This means that we have a lot of higher frequency shared haplotypes, so haplotypes that are spanning multiple locations, and then these are surrounded by many low frequency private haplotypes. However, for the majority, as you can see, the haplotype networks are shared across the locations, including one from Pritz Bay over in East Antarctica to the Antarctic Peninsula. So as I mentioned, because we had these such large sample sizes, we were able to look at their phylogeographic structure as well as look into the actual structure of species across populations. And we were able to conduct these listed statistic tests to detect demographic expansion, genetic structure, molecular variance, and molecular differentiation. And what we found, along with how these animals' structures were formed, was that for these 2 geographically structured species, we found this excess of rare or private haplotypes, which indeed to us inferred, along with the statistics we conducted, that there were historical population bottlenecks, and these organisms were able to locally diversify within their refugia before glacial melting and population expansion. Whereas for the geographically diffuse species, we found, as I mentioned before, these highly represented mixed haplotypes surrounded by many low frequency, closely related haplotypes. And as you saw, this animal was very widely geographically spread. So this indicated to us that these animals were able to continually diversify during glacial maxima. So what this says to us is that we have animals with the same life history traits, however, they had different refugia uses. So what this looks like, rather than using words, I figured we'll use pictures. So species 24 and 28, if you imagine the glaciers advancing out, these previously widespread species were then barricaded into these glacial refugia, where they were able to continually diversify, and then as the glaciers retreat, the organisms were then able to expand from there. Whereas for species 29, the glaciers would have advanced and this organism was somehow able to maintain distributions and maintain connectivity across the continent throughout time. For yes, as I said, a tiny organism, this is extremely perplexing, and there is really not many good examples of how these animals actually could get around in these times. We have a depth distribution of about 30 meters to 800 meters, and most of the Antarctic continental shelf is deeper than 1,000 meters. So there are a lot of unanswered questions, but looking into these first fine scale distribution patterns is a really great step forward in understanding this species complex. With that, I'd like to say, of course, a huge big thank you to my brilliant supervisor, Dr. Nerida Wilson from the West Australian Museum, and a few extra thanks to people who helped me out in the lab, Lynette, Mia, and Alex, and of course, all the funding bodies that helped for sequencing and lab costs, everything associated. Thank you all very much, and thank you, SASB, and thanks for listening.
Thank you for that, Paige. We're ready to move on to Lauren. Any questions for Paige can be sent through during the chat or put through to the panel discussion afterwards. Paige, I did have a quick question about your talk. So firstly, super cute animals. It always makes the work easier, but I was wondering how these nudibranchs are actually dispersing. Is it just a simple case of they're moving into the kind of strong currents? Are they able to raft into these different areas?
Such a good question. And it would definitely be a lot easier if I could just say, yes, they raft, and that makes everything brilliant. There have been records of rafting. In my slides, one of the pictures is actually the egg casing on top of a sea cucumber. So it's an accidental placement of eggs, but that would have facilitated some kind of further distribution. And there definitely has been records of egg masses being laid on bryozoa, for example, which has been recorded to be moved via anchor ice, which would have been dislodged and potentially settled elsewhere for rafting. But in terms of overall, we can't say that that would be our definite explanation as to how these slugs are getting around, because it just seems too far and few between, especially when your egg casings as well as the slugs are all pretty adhesively not that competent. So if one strong current or one big wave, they would just get lost and probably pushed into the warmer northern oceans and get lost and denature from heat. So in terms of how they actually get around, that's just the biggest question on all of our lips, because it just makes absolutely no sense.
Fantastic. Thank you for answering that.
Thanks for the question.
So I'm all out of questions.